of you. Today we are going to see about the detection of Clostridium Perfringens in food. Okay, first, what is Clostridium Perfringens? It is formerly known as Clostridium welchi or Bacillus welchi, and it is a gram-positive rod-shaped organism. It requires anaerobic condition. It is a spore former and it is a pathogenic one. And Clostridium perfringens present in the decaying vegetation and intestinal tract of humans, in other vertebrates, insects, and it is also present in soil and marine sediments. And it is naturally cause the food poisoning, particularly in the United States, and sometimes it won't cause any of the poisoning also. And infection, it indicate with the tissue damage by tissue necrosis, bacteremia formation, and emphysematous cholecystis, and gas gangrene. Gas gangrene is the main indication for this Clostridium perfringens infection, and it is also called as Clostridial myonecrosis. And that perfringens, it means burst. It is obtained from the Latin. It refers to the disruption of the tissue that mainly occurs during the gas gangrene. And the toxin involved in this gas gangrene is an alpha toxin. That toxin disrupts the normal cellular function by disturbing the plasma membrane in the cells, and it causes the gaps in the membrane. So it forms the abnormal tissue functions and cellular functions, and mainly it participate in the anaerobic infections. Of course, it is an anaerobic organisms. The treatment is a hyperbaric oxygen. So it has helped in the management of the clostridial tissue infection. It has completely detoxified the patients and relieved from the bacteremia also. And let's see this how it was detected in the food, Clostridium perfringens presence. So it was expressed in a small video, then afterwards I'll explain it. Okay, let's see the video for 1 minute. Yeah. Okay, so now we will see the methods. First, we have to use the buffered peptone water. Yeah, we have to detect the Clostridium perfringens in the food. So, which food sample is to be detected? That should be taken in the 10 gram, and we should be mixed in the 90 ml of this buffered peptone water. So, it is in the ratio of the 1:9. And after that, you are completely homogenized. Then we have to use the pour plate technique. So for that we are using the agar tryptosulfide cycloserine agar, that is the selective agar for Clostridium perfringens. So in the pour plate technique, first we have to add the 1 ml of sample and we have to pour the media on the top. This is the pour plate technique, and after the solidification of this we have to pour media on the top, solidified top. Then we have to incubate that plate in anaerobic condition at 37 degrees Celsius for 24 to 48 hours, and if it contains the black color colonies, it was denoted as the positive culture. Okay, so we are using the tryptosulfide cycloserine agar, or it is called as perfringens agar base. So in this we are observed the colonies, black color colonies. Okay, represents the Clostridium perfringens. Okay, otherwise if the food sample doesn't contain the Clostridium perfringens, means it won't come. This one. Okay. And further we have to confirm by means of three techniques. The first one, fluid thioglycolate medium, and the second one, lactose gelatin medium, that is LGM, and then third one is motility nitrate medium, MNM. So three techniques, the FTM, LGM and MNM. So first one, FTM, denotes the growth. So in this medium, after the preparation, we have to incubate at 37 degrees Celsius in an anaerobic condition for 24 hours, and after that, if we observe the growth of the organisms, then it denotes the positive one. So in this, we have to inoculate the culture that was taken from the plate. Okay, Perfringens, a hair-based plate. And then another one technique is LGM. So LGM also, after the preparation, it should be incubated at 37 degrees Celsius in an anaerobic condition, and in this, if it is positive, it was liquefied, and if it is a negative, it was in the solidified condition. And bubble formation and also yellow color formation also formed, that denotes the carbohydrate fermentation. So here we are observing this is called fermentation. Okay, so in after that also we have to keep this in refrigerated, and that condition also it won't get solidified, then it is confirmed as a positive Clostridium Perfringens organisms. And then MNM. Okay, MNM, in this we have to stamp the culture mainly for the motility observation. So here non-motile, the organisms grows where the organisms was stabbed, it won't penetrate into some other surfaces, that showed the non-motile nature, and it should be incubated at the 37 degrees Celsius in an anaerobic condition, then we will observe the results. So after that also we have to add the naphthalene reagent. So after the 15 minutes, if it forms the red color, it showed the conversion of the nitrate to nitrite. So nitrate presence also observed in this, nitrate production also observed in this medium. So that is MNM medium. So after that we have to go for further confirmation by taking 1 ml from the FTM medium and prepare the grossly milk medium. So this is mainly for the stormy clot formation. So for that we have to keep this in a water bath for 46 degrees Celsius for 5 hours. Within 2-3 hours also we will get the results, but further if we are increasing the time up to 5 hours. So this is the stormy clot formation. If it is forms, it showed the positive culture of Clostridium Perfringens. The further it was confirmed. So these are the some of the tests that are used in the industries to confirm the Clostridium Perfringens presence in the food, presence or absence in the food. And if you have any doubts or clarification, you can mail me at shivaza91@gmail.com. And then I would like to thank our senior Suresh and some other members for helping me in this presentation. And thank you all of you, and happy Dasara too.